Exploring gram staining, a comprehensive guide to bacterial cell wall classification. Introduction. This presentation provides a comprehensive guide to gram staining, a widely used technique in bacterial cell wall classification and microscopic analysis. We will discuss the principles, procedure, and applications of gram staining. Bacterial cell wall. Bacterial cell wall is a complex structure that provides shape, rigidity, and protection to the cell. Gram staining is based on the differences in the cell wall composition of bacteria. We will discuss the structure and function of the cell wall in detail. Gram staining principle. Gram staining is a technique used for differential staining of bacteria that helps in distinguishing them into two different groups based on the differences in their cell wall structure. The technique involves the use of crystal violet, iodine, alcohol, and safranin, each playing a crucial role in the staining process.
Crystal violet is used to stain the bacterial cells. Iodine acts as a mordant that binds the crystal violet to the cells. Alcohol is used as a decolorizing agent that removes the stain from some bacteria, and safranin is used as a counterstain that stains the decolorized bacteria. Gram-positive cell wall. The gram-positive cell wall is composed of a thick layer of peptidoglycan, which is responsible for retaining the crystal violet iodine complex during gram staining. This layer provides the cell with structural support and protection against environmental stressors. Additionally, the gram-positive cell wall contains teichoic acids, which aid in the regulation of ion movement and cell growth. Understanding the structure and function of the gram-positive cell wall is important in the diagnosis and treatment of bacterial infections, as it can impact the effectiveness of antibiotics and other therapies. Gram-negative cell wall. The gram-negative cell wall is composed of a thin layer of peptidoglycan and an outer membrane that contains lipopolysaccharides. This structure gives the cell wall its characteristic features and functions. The peptidoglycan layer provides rigidity and strength to the cell wall, while the outer membrane acts as a barrier to the entry of certain substances such as antibiotics and detergents. The lipopolysaccharides in the outer membrane contribute to the cell wall's negative charge, which helps to repel certain molecules and ions. The gram-negative cell wall is clinically significant because it makes these bacteria more resistant to antibiotics and immune system defenses. Gram staining procedure. Gram staining is a laboratory technique that consists of several crucial steps for its success. The first step involves fixation, where bacterial cells are attached to a slide through the use of heat or chemicals. The second step is staining, where the cells are covered with crystal violet, followed by iodine to create a complex. The third step is decolorization, which entails the use of alcohol or acetone to eliminate the stain from some bacteria. The final step is counterstaining, where a contrasting dye, such as safranin, is applied to the cells to display the decolorized bacteria. To obtain accurate results, it is essential to carefully follow each step and utilize the appropriate reagents and timing. Gram staining applications. Gram staining is a versatile technique that has many applications in microbiology, clinical diagnosis, and research. Some of the most common applications, including bacterial identification, antibiotic susceptibility testing, and epidemiological studies. Limitations of gram staining. Gram staining has some limitations, including false positives, false negatives, and variability among bacterial species. Conclusion. Gram staining is a fundamental technique in microbiology that allows the classification and visualization of bacterial cells based on their cell wall structure. We hope this presentation has provided you with a comprehensive guide to gram staining and its applications. Thank you for your attention. Thanks.